I think in terms of the fact that CRISPR-Cas systems can be divided, you have the functions of the different functionality, the stages by which the CRISPR-Cas system works: the adaptation stage, the interference stage, processing stage, and the fact that these stages are functionally independent allows the cell to recombine the functions in different combinations depending on the genome that you're looking at, right? So what does that say about the way that CRISPR-Cas systems evolve? I think that's a key question to be asked here. And obviously it's all speculation because we don't know at this point. But I think the fact that these subsystems of the CRISPR-Cas system can be recombined must mean that they're able to function independently. And so then the question becomes, can they function completely independently from each other, where you have an interference system carrying out a function independently, which does not necessarily have anything to do with immunity, with cellular immunity, and you have an adaptation system over here taking up new spacers, sampling DNA from its environment and inserting it into the host chromosome, independently of any immune function, right? Mm-hmm. Because the systems might have evolved independently of each other and then might have met each other by chance and turned into an immune system. Yeah. The modules themselves might not even be aware, you could say, so to speak, that they're actually working as an immune system because their baseline function is something completely different. And we're seeing indications of this, I think, in the studies that are going on at the moment and some of the data that's been coming out from some of the recent studies. I think there are indications in this direction. Just to give two short examples. One example is the way that the adaptation machinery works and takes up spacers against new viruses. It seems the adaptation machinery is not able to distinguish between host DNA and viral DNA, so that most of the spacers which are taken up at any given point are actually spacers derived from the host chromosome, which means that most cells, upon activation of the adaptation machinery, commit suicide. And so we see a stark decrease in the growth rate of the cells when adaptation is enabled, right? And so the only cells that end up surviving are the ones which have taken up spacers against the virus, right? So that's an indication that the cell is not really, that the adaptation module is not really tailored for immunity. If it was, it would have had a better mechanism for distinguishing between self and non-self DNA, right? So that's in terms of the adaptation. Another thing that we actually found here in Copenhagen is that we see that type 3 systems in particular, which are often found disjunct from any adaptation module. You have a type 3 module, interference module, which is often far away from any CRISPR-Cas systems which are present in the cell. And so then again, you would ask, maybe this type 3 system is carrying out some different function beyond immunity. And what we see apart from that is that the type 3 systems are often flanked by additional genes, all kinds of different genes, which co-evolve with the type 3 system. So we know that those genes have had a long history of following the type 3 system. They must be carrying out some function. When we look at the genes, some of them encode CARF domains. The CARF domain proteins have already been shown to have an immune function. So, but some of the other proteins that we're seeing encoded are like transmembrane domains, proteases, nucleases, helicases, all kinds of different proteins. And while those proteins might have an immune function, it's also conceivable that those proteins help the type 3 system to interface with other parts of the cell carrying out other functions, right? Because the type 3 system, interference, nucleic acid interference has a lot of potential applications beyond immunity. It can have applications in, regulation of genes, for example, regulation of replication, other things like that. So I think, that's the answer to that question.